Hi, my name is Siddharth.
Hi, this is Sunny.
Hi, my name is Sophia. We are Group 12, and we are studying the effects of adenosine, antidiuretic hormone, or ADH, and histamine on fluid balance of frog systemic circulation. Adenosine is a nucleoside involved in many metabolic pathways and is released from cells as a result of ATP usage. Adenosine can act as a signaling molecule for the cardiovascular system.
ADH is an antidiuretic hormone that acts as a signal in the kidney to reabsorb water.
Histamine is a chemical messenger that mediates immune responses, stomach acid, and allergy symptoms.
A frog's systemic circulation was perfused with Ringer's in 3% dextran solution via catheterization of the left aortic branch. Most internal organs were removed, excluding heart, lungs, and kidneys. The frog was suspended by an isometric transducer to measure change in weight. The isometric transducer was connected to the transducer power supply. The power supply was connected to the positive input on the preamp, and the preamp output was connected to channel 1 positive input of PowerLab. Flow rate was measured by counting drip rate, which is the number of solution drops rolling off the toes of the frog per minute. Calibration of the isometric transducer was done using the consecutive addition of 10-gram weights to measure the relationship between tension and change in voltage. The relationship was described as a calibration equation and determined to be change in tension in grams is equal to 0.170 grams per millivolt times the change in transducer output in millivolts. During the frog perfusion experiments, data from the isometric transducer was recorded as transducer output in millivolts and time in seconds. The raw data depicts a change in millivolts over time. Using the calibration equation, we converted the slope between two data points from millivolts per second to grams per second. An example calculation is shown here.
Vascular smooth muscle, which is the smooth muscle surrounding vessels, has an important role in controlling blood pressure and flow through the vessel. When the smooth muscle contracts, there is vasoconstriction, and when the smooth muscle relaxes, there is vasodilation.
There are three main transduction pathways in smooth muscle: GS, GI, and GQ. The net effect of the GS transduction pathway is relaxation of smooth muscle. When the alpha subunit of the GS protein gets activated, it can activate adenyl cyclase, or AC. AC is an enzyme that catalyzes the conversion of ATP into cyclic AMP. Cyclic AMP can activate protein kinase A, or PKA, which is another kinase that can phosphorylate substrates. PKA can phosphorylate myosin light chain kinase, or MLCK, to inhibit it, and it can phosphorylate myosin light chain phosphatase, or MLCP, to activate it. MLCK phosphorylates myosin using phosphate from ATP, while MLCP cleaves the phosphate off myosin. Dephosphorylated myosin has an inactive ATPase, meaning it can form cross bridges with actin, leading to relaxation of the smooth muscle. PKA can also phosphorylate potassium channels to activate them, leading to more potassium efflux of the cell, and thus a more negative membrane potential, turning off L-type calcium channels. Therefore, the net effect of the GS pathway is relaxation of smooth muscle. Furthermore, when the alpha subunit of the GI protein gets activated, it inhibits adenyl cyclase, which would ultimately cause the net contraction of smooth muscle through the same mechanism. In the GQ transduction pathway, a ligand first binds to a receptor. Then the alpha subunit of the GQ protein dissociates and activates phospholipase C, or PLC. PLC then converts phosphatidylinositol biphosphate, or PIP2, to inositol triphosphate, or IP3, and diacylglycerol, DAG. IP3 binds to the IP3 receptor on the sarcoplasmic reticulum, which causes the calcium ions to flow into the cytosol. DAG activates protein kinase C, or PKC, which activates L-type calcium channels and inhibits potassium ion channels, allowing more calcium ions to flow into the cytosol. Free-floating calcium ions activate calmodulin, which activates MLCK. MLCK phosphorylates myosin light chain and causes vasoconstriction.
Starling's law of ultrafiltration decomposes the net movement of fluid between the blood and the interstitial tissue. The capillary blood pressure, or PC, is the hydrostatic pressure inside the capillaries, pushing fluid from the vessel into the tissue. The interstitial tissue pressure, or PI, is the hydrostatic pressure of the fluid in the tissue, pushing fluid from the tissue into the vessel. The interstitial tissue oncotic pressure, or PI, is when the interstitial tissue develops an osmotic pressure drawing water toward it, such as through ions in the tissue. The capillary oncotic pressure, or PI-C, is when the fluid in the capillary develops an osmotic pressure drawing water towards it, such as through cells and proteins like albumin in the vessel. K, which is the filtration coefficient, defines how permeable the blood vessel is. This ultimately gives us Starling's law of ultrafiltration, where if phi is increased, the change in frog's weight is increased because there is more fluid going from the vessel into the tissue. When phi is decreased, the change in frog's weight is also decreased because there is more fluid going from the tissue into the vessel. Ohm's law defines the flow rate through the frog's blood vessels. Ohm's law says Q, which is the flow rate, equals the change in pressure across the tube, delta P, divided by the resistance. The resistance is further defined as the equation 8 times eta times length divided by pi r to the fourth, where eta is the viscosity, defining how viscous the fluid is, L is the length of the tube, and r is the radius. In this experiment, eta and the length of the tube are not going to be changed, so really the radius is the only major factor. And when the radius of the tube is decreased, the resistance of the tube is increased. The recorded drip rate when the frog was perfused with Ringer's and 3% dextran was 60 drips per minute. And when the perfusate had 2 millimolar adenosine, the recorded drip rate was 64 drips per minute. The change in frog's weight over time in grams per minute was 4.8 E-2 when just Ringer's and 3% dextran was the perfusate, and when adenosine was in the perfusate, the change in frog's weight over time was 7.53 E-2 grams per minute. Vascular smooth muscle has A1 and A2 adenosine receptors. A1 adenosine receptors activates the GI transduction pathway, while A2 adenosine receptors activates the GS transduction pathway. From the data, it is predicted that A2 adenosine receptors are the main receptor type being activated by adenosine. If A2 receptors are being activated, vasodilation occurs in the frog's vessels, explaining the increased flow rate due to a lower resistance. Also, PC, or the capillary blood pressure, would be increased when vessels dilate. This causes fluid to go from the frog's vessels into the tissue and causes phi to increase. This explains the increase in the rate of change of frog's weight observed when adenosine was added to the perfusate. This also makes sense physiologically because A2 receptors have a lower affinity for adenosine. Because such a high concentration of adenosine was used in this experiment, the A2 receptors would be fully activated, despite their lower affinity for adenosine.
The drip rate when Ringer's with 3% dextran was perfused was 56 drips per minute. When 1 microgram per milliliter of ADH was added, the drip rate was 52 drips per minute. The change in frog's weight over time when Ringer's with 3% dextran was perfused was 0.1557 grams per minute. When 1 microgram per milliliter of ADH was added, the change in frog's weight over time was 0.0627 grams per minute. ADH can bind to vasopressin 2 receptors, which would trigger the GS transduction pathway. As discussed, the GS transduction pathway ultimately causes vasodilation of the frog's blood vessels. However, our frog expressed more vasopressin 1 receptors in vascular smooth muscle, which resulted in the activation of the GQ transduction pathway, leading to vasoconstriction of the arterioles. Vasoconstriction caused the radius of the arteries to decrease. The decreased arterial radius lowered the capillary pressure, and using Starling's equation of ultrafiltration, a decrease in capillary pressure also decreases phi. This caused more fluid to leave the body, which is why the frog's change in weight over time decreased. Using Ohm's law, the decreased arterial radius would increase the resistance in the blood vessel and lower the overall flow rate, resulting in a lower drip rate.
The drip rate when Ringer's with 3% dextran was perfused was 60 drips per minute. The drip rate when ringers with 3% dextran with 1 millimolar histamine added was 56 drips per minute. The change in frog's weight over time when ringers with 3% dextran was perfused was 0.0147 grams per minute. When ringers with 3% dextran with 1 millimolar histamine was perfused, the change in frog's weight was negative 0.0677 grams per minute. The binding of histamine to H2 receptors initiates the GS transduction pathway. As described earlier, the initiation of the GS pathway results in vasodilation of vessels in the frog's systemic circulation. The binding of histamine to H1 receptors in smooth muscle activates the GQ transduction pathway and results in vasoconstriction. Because we observed an overall decrease in frog drip rate and decrease in frog weight in the presence of histamine, there was a dominant presence of H1 receptors in the frog's vessels compared to H2. This is further supported by Starling's equation of ultrafiltration and Ohm's law. Starling's equation of ultrafiltration and Ohm's law describes vasoconstriction of the arterioles to decrease PC, causing increased fluid from the frog's tissues to move into the capillaries. This is why we observe a decrease in weight. Due to the reduced radius in arteries that caused by vasoconstriction, the total peripheral resistance increased and the flow rate within the blood vessels decreased.